right, so next we have Ellen with gene manipulation of the extended phenotype of Gall-inducing insects.
I'll get answers. I can be off with that.
We have the same plan.
Probably it's like the cheapest one on Amazon. Yeah.
So do you have mine or yours?
Mine has my missing or my chunk missing. All right. So we're moving away from death and dying to try to get at solving a biological mystery and maybe learning a little bit of natural history. So who here has heard of a plant gall? Great, couple in the room. But okay, so that's what we're going to talk about today. So plant galls, see if got it. It's clicking onto yours, not mine.
Yeah.
Funny, now I see what you're saying.
No, I think maybe just do that.
Great. Okay, so a gall, let me see if this will work. Yep. A gall is an abnormal growth on a plant. So this is a gall right here. And galls can be induced by a variety of organisms. So bacteria, fungi, mites, nematodes, and then insects. But insects form kind of the most structural galls, and this is why I like to study them. And the reason why a gall exists is that it provides nutrition to an insect's offspring and it protects them from harm. So if we look at this little dude right here, so this is a slice of this gall here. You can see that there's these individual chambers. Inside is the larva of a wasp right there. And surrounding that little larva is this nutritive tissue. So it's this lipidy, starchy, nice stuff for that larva to eat, and it helps it develop. And then around it is this very, very dense tissue that tastes bitter. Trust me, I tried it. And it protects that from parasitoid wasps or rodents or anything else that's trying to get in this little protein packet here. So the reason why galls are cool and I like to study them is that this structure is made out of plant genes and plant proteins and everything like that. But the function or the shape of it, the timing of it, everything about this gall is dictated by the wasp that is inducing it. So this is considered to be an extended phenotype. So it's wasp or insect genes that are actually dictating the morphology of a completely different organism by manipulating that genome. And so this is kind of an interesting thing. So not only does the insect have to evade host defenses, so similarly to any other herbivore, it also has this extra step of co-opting control of plant development and forcing that plant to then rechange its entire developmental program to make this unique structure. So there's about 210,000 species of gall-inducing insects. It's evolved at least a dozen different times. It's a very popular kind of evolutionary intervention. And they come in very, very diverse shapes and sizes. So this one here, this one here, this one here, this one here are all on oak trees. So I don't know the last time I saw cotton candy on an oak, but these insects can make oaks do it. They can also do very interesting things. So this one right here, Dishvolcastus cinerosa, it actually, that gall kind of looks like a peach, but it secretes nectar. And so the last time an oak tree secreted any sort of nectar was about 100 million years past to something, because they don't have nectaries or flowers or anything like that. And then that nectar actually attracts ants that protects the gall from parasitoids and stuff like that. So these interesting tritrophic interactions. So even though I hope by this point I convince you that galls are astounding and amazing, but where the biological mystery comes in is that no one knows how the insect does this. So we have very, very little clues about how they can do this. There's been very little studies genomically looking at them. Currently to date, we have only connected a single gene in an aphid to any sort of gall phenotype. And so that's kind of what we're kind of set up to do in my lab, is to try to figure out mechanisms behind these insect galls. And one of the major problems of why we haven't been able to solve this yet is that it's really difficult to find a model system for galling. So they work in these kind of hard systems like oak trees and willows. They tend to have very long generation times, all these things that don't make great models. But I think in my lab in the last 5 years here at the University of New Mexico, I've been able to kind of come up with a couple of systems that can get at that questioning kind of sideways way. So the first one is the blueberry stem gall wasp, Hemidus nulipennis. And this is the gall that we've been looking at through the introduction here. This is what the little waspy looks like. And the reason why, and this is with collaboration of my graduate student, Adam France, and then also my collaborator, Glenn Hood at Wayne State. And one reason why I like Modest so much is that it's an independent evolution of galling. So the vast majority of galling wasps are either cynipids that gall oak trees or sawflies that gall willows. But the Amanus is actually here in a large group of just parasitoid wasps. So these are surrounded basically by parasite predators, and they have kind of switched their life cycle to sting plants and manipulate them. So we can kind of compare that to what's known to the other ones to see how these unique innovations can happen. So this is the life cycle of these wasps. They oviposit in the spring, and then about 15 days later, a swell starts that could be either pink or green. Around 80 days, that is the mature gall, and that stays throughout the summer and fall and winter. They oviposit in the, or they overwinter in the gall, and then in the spring they emerge as adults to start all over again. So one generation a year, that's part of the trouble. So what we wanted to do first was to ask, how does a wasp influence the expression of its host? And does the gall mimic any other tissue in the plant? Because this is like blueberry-esque, right? And then that was kind of one of the theories that they might be modified fruits or flowers. So to do this, we did transcriptomics. So we sequenced every gene that was being expressed at that current moment. So these things that are about to turn into proteins, so kind of can tell us what the plant is planning to do. And so we did it for each of these stages. And then the mature gall, we separated out that inner nutritive tissue from that outer defensive tissue. And then we paired these with matched non-gall samples. So to match sunlight, wind, water, all the other stressors that might be happening. And then we also sampled a bunch of normal tissue. So fruits, flowers, stems, roots, anything we could kind of get our hands on. So this is a principal component analysis. It's showing of the entire gene expression profile. Basically, if two dots are close together, their expression is very similar. If they're further apart, they have very different expression. So we're first looking at the normal tissue. So we have our fruits, we have our roots, we have our stems and leaves that are kind of clustered together. And so if we map on those beginning stages of the gall, right after oviposition and that swell, we can see that they actually cluster pretty well around the area of the things that it was derived from. So they look like a modified stem, right? But even though they are closely resembling to stems, we can also find some really interesting genes that are not supposed to be expressed at the time. So one is a spidosulfokine, which actually de-differentiates cells. So a cell that is a leaf cell can de-differentiate down back to a meristemmal cell again. You can see that might be a really great idea to kind of wipe the slate clean to start their new developmental program. We also have these MADSBOX proteins. So MADSBOX are these transcription factors that basically lay out the blueprint of organs. So there are flower MADSBOX, there are fruit MADSBOX, there's root MADSBOX, and there's stems. And so in galls, they have a weird combination of expressing both stem MADSBOX as well as fruit MADSBOX at the same time, which does not happen anywhere else in normal development. And as far as we can tell, it hasn't been seen in the plant before. So, and then, so if we then look at this mature gall, we can see that they're in no man's land. So the mature gall looks nothing like any of the other tissues on the plant. About 30% of the genome is differentially expressed between that inner tissue and the tissue that was derived from the stem. So we can really see that this is a parasite-specific gall. So if we kind of look at this in a slightly different way, this is the number of significantly differentially expressed genes from a gall plant to their matched controls. And very cool, but very frustrating to me, is only about 0.3% of the gene set has consistent expression through all of gall development. So there's no such thing as just a gall profile. This is just a dynamic, ever-changing, developing organ with multiple tissues that is going to be exceedingly complex to work out, but I'm up for it. The other reason why I like Modest is that currently they're at outbreak densities in northern Michigan. So these are different varieties of blueberries, and Jersey, the one that gets hammered the most. This outbreak started around 2016 and has continued up to today. So my collaborator asked a farmer to send us some gall samples, and he sent us this. Cool. And another example is my collaborator was in the field with his graduate student and couldn't see him 50 yards away because of the density of wasps in the air. So this one that's hammered really the worst is actually the most planted with 32% of all Michigan blueberries. So we kind of need to figure it out. So one thing we wanted to do is we looked at the entire plant expression, but I don't have time for that right now. But what we did also look at is the venom gland. So the venom gland is this organ that that is used to sting the plant at first and usually probably mitigates the immune response of the plant. So it kind of just dictates the susceptibility versus resistance of the plant for this egg and the beginning gall development to happen. And what we see is compared to completely native wasps, so wasps that are on native blueberries, the outbreak wasps are significantly different from that, and also significantly different from wasps that are on other just domesticated non-outbreak wasps of that. So we can actually start looking at these genes that are the difference between these two and kind of get at a little bit of outbreak dynamics and what changed over the short period of time to make it so that they were so much more successful on these crops. Yeah, so outbreaks with 3 times more. You can also see the same thing in larval expression, but it's a little bit less. So Hemidus is great for galling evolution and outbreak dynamics, but I also wanted to talk about another system to which you guys can see pretty much in your own backyards here. So this is Assyarina, and this is a system that I have started with Vince Martinson, who is also an assistant professor in the biology department, and I guess my husband, and then also my absolutely newly minted Dr. Quinn Bain. She just attended like 2 weeks ago. So these two are Assyarina trixa and Assyarina biglovia, and they form galls on Ericameria nauseosa or rubber rabbitbrush or chamiza. So this is a plant that you see usually along the highway that blooms yellow in the fall, right? And you can see it around campus too a little bit. And so these guys form galls on that. So trixa forms this very smooth resinous gall, and this is the one you can see on campus. So if you even just go straight out here on Martin Luther King, right outside the building, there's a bunch of rabbit brush right at the intersection of Martin Luther King, and they're covered in galls. So you can go check those out, and you'll see them everywhere now that I've induced you to gall searching behavior. And then Assyarina biglovia has this very cottony gall. And while they make these 2 different galls, they're also separated by their host plant. So it's all the same species, but Ericameria nauseosa has 23 varieties. And so these guys specialize on different varieties of that host plant. So again, here's our 2 species, and this is the ranges of their host plant. So the smooth galls are sitting in this smaller thing. The gray galls have a much larger range. However, if we map on all 23 varieties, it covers the West. And if we look at what these different morphologies are, is that we have a kind of a really huge range of different morphologies that these flies do, from basically having no external structure and finials and cottony things and stuff like that. And the flies have little different patterns on their wings that are fun to figure out. So we wanted to know, how are these flies actually related to each other, and then how are they evolving with their host plant? So we ran around and collected all of the colored circles here with paired flies and plants. And then we also gathered a bunch of other plant data that was from a previous thing that had unknown gall associates. And we made a tree. So this was done through reduced genome representation, so dd-rad data. So we basically randomly sampled a lot of small chunks of the genome and put them together. So this is around 6,000 individual genomic SNPs making this. And it ended up pretty well. So the flies have very good support with this, but it ends up being they're pretty young. So this node right here is only about 30 to 60,000 years ago. So here are the 2 I've been talking about so far, biglovia and trixa. They were originally thought to be sister species before we started doing this. And then we have kind of our outgroups that made sense. So opaca, the one without any external gall, is sister to the rest of the clade. We also have, and then we have these 2 that we thought were going to be different species because they have a very different morphology between this cottony and a finial kind of thing. But when we look at the clade, they are all interspersed together, so we can't really tell them apart genomically. However, if we do just kind of a sliding window and looking across it, and see if there's like smaller chunks that are sorting one to the other, we actually do find a couple regions of the genome that seem to be kind of segregating between these 2 groups. So we might be able to figure out some of these galling genes by looking at these guys. Then we also had a secondary trixa Trixa that we thought was going to be the same as this Trixa, but ended up not being the sister species, but one even beyond that. That's kind of specialized to red dirt around Moab. And we were able to officially describe a new species. So this is Acerina luminaria. And so we named it luminaria because the gall is actually the shape of a candle flame. And you can see it very well in the winter when the leaves drop. And so it's very reminiscent of our New Mexican tradition of having luminarias around Christmas. And so that's how we got this name. So we wanted to know basically, so now we know how the flies are related. So are they actually co-speciating? So are they following the phylogeny of the plant, or are they kind of doing things different ways? So we had to figure out the plant genome first. So we did that. It's a lot more messy because we're looking within one species again, right? So we're only looking at varieties. But we were happy to see in this particular phylogenetic analysis, we were able to actually pretty closely replicate what was kind of known as the varieties through morphology. And then we put the two together. So as you can see, this is called a tanglegram, obviously. And it is quite tangled. So we found out that actually co-speciation is not actually what's happening. The major driver for the evolutionary force of this is host switching. But we have a lot more to kind of learn and put together. But as we were figuring all these things out, we were also able to kind of find a little bit more kind of crazy natural history too, which I will go through right now. So we've only been talking about the inducer and the host plant. However, I mentioned parasitoids before a couple of times, so there's those, but there's also a bunch of other things. So galls are considered to be ecosystem engineers because they create a novel environment which other organisms can actually use. And with that, they can increase the heterogeneity and species richness of an area. And so they end up being these hotspots for biodiversity. And so we were wondering, okay, who's actually associated with these two? So we collected a bunch of stuff. We had everything reared out of it, and we found a bunch of stuff. So we found 22 different insect species that are associated with just these two species of Acerina, including this adorable weevil. And there was some of the galls, the cottony galls, that had kind of a weird texture on them. And when we peeled back the cotton, we found galls on galls. So these are hypergalls, which coined that term. And these are induced by a little midge that look like a Cheeto. That causes these galls to form on the surface of the big lobia gall. And so do they have a parasitoid community? Yes. So 7 species of parasitoids were found associated just with a hypergall and not with the primary inducer at all. So that made it so our record was from a single gall, we reared 102 arthropods, which means that is equating to 194 arthropods per cubic centimeter. So these things, I'm not lying, we're not like making stuff up. These really are biodiversity hotspots. So we had this amazing trophic web that we were able to describe. And overall, the system has been absolutely a sandbox of fun to play with for connecting insect genotype to gall phenotype, learning about speciation and community assembly. And then overall, basically my lab is just using different systems in unique ways, integrating both their evolution and ecology to try to understand the unique and the common mechanisms behind gall. And with that, I would like to definitely thank all of my people and especially the CARSI team for facilitating all of these genomic wildnesses that we do. Okay, thank you. I don't have any questions. Oh, sorry, Daryl.
So do I understand? So you have like parasitoid wasps that do this,
Mm-hmm.
and you have flies that do this,
Mm-hmm.
and they're like not really dull. There's no. Does that like indicate like it's like some sort of proteomic thing or small molecule signaling or something?
Yeah, no, I mean, very good question. Like the only like hint that we have right now is that like that one gene that is known in aphids seemed to be kind of like a something that is binding directly to the DNA. What it's doing, be it like a transcription factor or something like that, we don't know. So yeah, really good question. No idea. Lots of places we can look. So yeah.
Thanks. Yeah, JJ had a question.
Yeah, sorry, probably a stupid question. But when you're collecting these goals, I think about the amount of text and we're doing all the different flies. So you have to wait for the fly to emerge, or just what time do you harvest the gall to get your specimen?
Yeah, no, not a dumb question at all. Has taken us years to get this down. Not great, but good enough. But yeah, you basically have to dictate it to the biology of the fly. So we have to wait until they pupate to pluck it off. Otherwise, then the signal crosses will get mixed up and the fly will die. And if you prematurely kill the inducer, the gall will stop growing. And a lot of times the nutritive tissue reverts back to kind of relatively normal plant tissue. So it is active signaling happening.
Any other questions? All right, we're about half hour late because I am the worst person to be cutting great research off. I want people to talk for longer and longer and longer, but thank you very much for that presentation. Now we have coffee.